Assalamu alaikum. I am Sadia and today we are going to conduct an experiment, that is examination under a microscope an animal cell and a plant cell using an appropriate staining technique such as iodine or methylene blue. For this purpose we will use onion epidermal cell as a plant cell and frog's blood cell as an animal cell. We require some of the materials such as onion cover slips, a stainer such as methylene blue. You can also use iodine instead of methylene blue, a watch glass, a glass slide, dropper, water and a microscope. You need to cut the onion in a vertical manner and then you have to take the thick layer present inside each layer. Take this and wash it with water. Take the onion epidermal and wash it with water. You can use 1 or 2 drops of water to rinse this in this manner. After rinsing it with water, you need a stainer such as methylene blue or iodine, and you will use the stainer to stain this onion epidermis. Then after this, you will transfer this onion epidermis to glass slide. In this way, and then you will use a cover slip to cover the onion epidermal. Now your slide is prepared, and with the help of a microscope you will observe the onion epidermal cells under the microscope, and then you will make the observations. When you will see the onion epidermis under the microscope, you will be able to make some observations, such as you will see that cell wall is present and there will be a large central vacuole present, due to which the nucleus is not present in the center but it is present on the side. Then you will see large number of organelles, which are also known as subcellular structures. This will demonstrate that the onion epidermal cell is an example of an plant cell. Then for an animal cell, you can use frog's blood cell or a human cell, a human blood. For this purpose, for an animal cell, you can use frog blood or a human blood. For this experiment, you can take a glass slide and place human blood or an animal cell on the slide and observe it under microscope. When you will observe an animal cell under the microscope, you will be able to make different observations, such as there will be present some blood cells and the plasma. Now the question is, what is plasma? Basically, plasma is the colorless fluid part of our blood, which consists of water, some soluble proteins. On the other hand side, there are 3 different types of blood cells present in an animal cell, such as red blood cell, white blood cell and platelets. Red blood cells are nucleated and oval in shape. They are red in color, and the function of red blood cell is to transport gases such as oxygen with the help of hemoglobin protein. Moving towards white blood cells, the white blood cells are irregular in shape. They do not have any specific shape, and they are comparatively smaller in size than red blood cells. The function of white blood cell is to defend the body against the invading microorganisms or diseases. Last, platelets are present, which are disc-shaped and they are even smaller in size than red blood cells. Their main function is that platelets help in blood clotting. No cell wall is present in animal cell as it is present inside the plant cell. So after observing these two, you will be able to compare an animal cell with a plant cell. Thank you.